So next up, we have Mohammad Taha Toghani, who is a PhD student at Rice University. Who'll be talking about estimating drug sensitivity on pancreatic cancer as a cold start problem in recommender systems.
Hello, everyone. Can you hear me? Sorry. Hello, everyone. Thanks for being here, and I hope you are all having a very good day. I'm going to talk about my work on estimating drug sensitivity on pancreatic cancer as a cold start problem in recommender system. This is a joint work with Dr. Kim and Dr. Jiang from UTHealth. Let's have a quick review of definition of drug repurposing. Drug repurposing or repositioning is the act of finding a new therapeutic use for an already approved drug. Let's take a look at the timeline of drug discovery for both traditional and drug repurposing processes. In the traditional method, as you can see in the first row, there are 5 stages, and as you can see for drug repurposing, the preclinical trials and phase 1 trials have been skipped. Since they are actually the drug is already approved, and this process have been ended in the first stage of drug discovery. So therefore, drug repurposing has 3 main stages. The first stage is to find a target population of patients for a drug, and then phase 2 and phase 3 of clinical trials. Phase 2 and phase 3 are FDA approval and review, and phase 3 is FDA post-market screening. So a good question is why drug repurposing and why we are interested in that. First of all, developing drugs can cost more than billions of dollars, and it is good to know that 90% of drugs being studied fail in the initial trials. In terms of time, as you saw in the previous slide, it may take more than 10 years to develop a drug. And now that we are in the middle of COVID crisis, it can save us a lot of time. And in fact, it has attracted a lot of attention to itself. Drug repurposing has lower risk and higher reward. In fact, due to the high risk of traditional drug development, big pharmaceutical companies are less interested in drug development for disease that can affect relatively a smaller population. Thus drug repurposing is a crucial method for orphan disease. And a good question is why computational approaches and why data science? Actually, these type of approaches give us a high throughput drug screening by automatically estimating drug sensitivity based on genomic data and pharmacological data. That can decrease the time for discovery of a target disease, and then phase 2 and phase 3. And the last question is why pancreatic cancers are important for us. Pancreatic cancers, despite their prevalence, which is really low compared to lung and breast cancers, they are deadly and incurable. And again, compared to common cancers, and their cell lines have been less studied. And their mortality rate is very high. For example, the chance of recovery for pancreatic cancer in the first year is less than 20%, and it can be decreased to 6% on average for 5 years. So what is our goal now? Our goal is to use drug repurposing to detect effective drugs that are inhibitive on other type of cancers for pancreatic cancer. And how we do that? Let's first see our dataset. Our dataset has 3 main parts, and it is provided by Second Healthcare Dataton from School of Biomedical Informatics at UTHealth. The first part is the relative inhibition of drugs on cell lines. There are roughly 1,900 unique cell lines of 14 different tissues, and we know Also, we have 44 pancreatic cancer cell lines that we know they're sensitive drugs, but we use it as ground truth for the performance of our method that we will explain later. And the number of unique drugs in this dataset is roughly 4,500 unique drugs. And from this pair of drugs and cells, we only know 7% of relative inhibition values, which is 1 if if a drug is sensitive on a cell and 0 otherwise. Thus, it is a very sparse matrix. Now, let me explain why we consider a cold start perspective for recommender systems. As you can see here, assume that cell lines be users and drugs be items. Then, for pancreatic cell lines, which we don't know the proper drugs for them, and we want to rank drugs based on the information of trained dataset, which is the cell lines of other cancers. We need to use this information, and that pancreatic cancers can be interpreted as new users in our scheme. Then drug repurposing would be cold start condition when we don't have any RI value here. And to do that, we need some contents about cell lines and drugs or users and items in cold start perspective. And what are those characteristics? That is the second and third part of our dataset. The second part of our dataset is the genetic expression for cell lines, which is based on FPKM. That is an accurate quantification of cells genetic, and it has, of course, some missing gene expression, and it has a sparse representation. And for drugs, what do we have? For drugs, we have their target genes with a binary number, whether if a drug it can target a gene, it is 1 or 0 if it is not, and Maxius fingerprints, which counts the number of 166 structures in drug, like, for example, oxygen structure or so on so forth. And it is also a sparse representation. So before explaining our method, let me talk a little bit about more about cold start problem solutions. There are usually 2 types of solutions. First one is content-based filtering, and the second one is collaborative filtering. In content-based filtering, we need to know some content about both users and items. In our problem, it can be the genetic expression, genetic target, and fingerprint. And the assumption in content-based filtering methods is that pairs with similar patterns in their content, by pairs mean drug and cell pairs or item and user pairs, with similar patterns in their content have similar output, which in our case is really relative inhibition, which is 0 or 1. And thus to detect this pattern, we need to use a machine learning model we can employ a machine learning model to recognize that pattern. That would be the content-based filtering. And for collaborative filtering, we need to know some content about users. For example, the genetic expression of cell lines here. And the main assumption here is that users with similar users have similar preferences. And how do we characterize quantize this similarity? We can use a kernel function and apply it on contents of users to quantify their similarity. And calculating the similarity between users and interpolating their preferences on which is RI values, we can suggest RI values to new users, which are pancreatic cell lines. So based on that, let me explain our method. This is an overview of method which has 2 main modules, pre-processing and prediction. For pre-processing, for each pair of drug I and cell J, we have the genetic expression, target gene, and fingerprints of this pair to be compressed by different deep autoencoders, and to reduce their size to a much smaller vectors. And we behave this obtained information as the content of the pair, and we use this content for pairs with known RI value and train a model that and it would be our content-based filtering, and that gives us some score based on content and RI value of known pairs. And for collaborative filtering, we use the genetic expression of all cells, and by the kernel, we characterize their similarity, and using some function which can be as easy as a weighted average or as hard as a graph neural net, we can quantify a score for new users for different drugs. And we add up these scores to have a final score that we call it our hybrid model, with some weights that can be determined by cross-validation. Thus, by now we explained our hybrid model, and of course there are a lot of technicalities in our method that are listed here. For example, how we handle missing value, how we handle cross-validation, how we compress the contents, and how we select models and kernels. There are simple and complex examples of each here. And just for cross-validation, note that if we select cell lines of a specific type of tissue, then we can make our model more to increase the generalization of our model, in the sense that it has not seen the cell lines of the same type in its trained data, like what it is for pancreatic cell lines. And for results, for each cell line and for each test cell line and cross-validation cell line, we introduce top 20 drugs with highest predicted scores, and compute the NDCJ factor on them, which is normalized discounted cumulative gain. Discounted cumulative gain is the sum of weighted relevance scores in the top 20 ranking list. And based on the and since it is normalized, and the maximum of it would be 1. And our method obtains 0.67 on this factor on pancreatic cancer cell lines, and it won the first place in SBMI second healthcare data. Let's see the sensitivity of our results. As you can see, for each different cell line in pancreatic cancer, our method could detect more than half of sensitive drugs for almost all of pancreatic cell lines, actually, except for these few cell lines. For most of them, it could detect more than half of sensitive drugs, and it is among the top 20 list, which is less than 1% of all drugs. And that is a promising result, in the sense that we can suggest a short list of drugs for each cell line to specialists that they can do the further experiments and investigations. With that in mind, I'm going to review some remarks of our algorithm, and then I will end. First of all, we formulate our problem as a cold start problem, and we develop a hybrid solution for that. And note that for our problem, collaborative filtering has a better performance usually. But why content-based filtering is important in our algorithm? Actually, content-based filtering helps incorporating abundant drugs. By abundant drugs, I mean drugs that are already approved past the preclinical trial and phase 1 clinical trial, but they have not been shown to be effective on a disease. So maybe their importance or sensitivity could be shown on new disease that we are going to discover by our computational approach. And there is a vast line of future works for our research, like scalability for online learning. Our approach is currently scalable, but on the number of cells and drugs. But since it is it depends on the sparsity of the joint matrix, but we want it to be scalable in the sense that it can take new trials online without need to being trained from the scratch, and use the information of that trial. And also, we are interested to employ some active learning mechanism that guide specialists for their future experiments. By that, I mean, say a specialist which pair of drug and cell have that their RI value is not known, which will have the most effect on our computational approach and help them to select the proper pair to do experiments on. And finally, an interpretable solution can help specialists in the sense that they can discover the unknown factors about cells and drugs. In fact, applying some constraints on the maximum level of sparsity on autoencoder rates can clarify the interaction between features in drugs and cells. And that is one way to increase the interpretability of our current solution. With that, I'm going to end, and thanks for attention. I would be more than happy to if I could answer any question.
Thank you very much. That was really interesting. One super quick question. So, of the 44 cell lines that were you were testing on, how different were the drugs that were found? Was it the same drug that was found across all of them, or was it a mix?
Yeah, basically. They were, I cannot recall very well, but yeah, some of them were diverse, and they were not exactly the same drugs. Yeah, and that's the promising part of our result.
Okay, good. So is that what you would expect, or would you expect them to act the same?
Actually, I'm not quite familiar with the medical side of the problem very much, but yeah, I think since we knew the ground truth for each cell line, so that we expected that this happens.
Gotcha. Great, thank you. All right, so we'll move on to our next speaker.